Yeah. So it's my job.
Okay. Oh, is that what this does?
Yeah. Focus out, focus in. Nope. I don't know if I like those. Alright. Come wide. Oh yeah. Look at this one.
That's a really pretty Chrysogorgia. This is possibly Chrysogorgia geniculata, or a bottle brush coral.
You want to push in on the...
There's a lot of squat lobsters in there, yeah, so let's take a look.
Alright, push in there.
There's a whole family of squat lobsters and a bunch of amphipods. This is a really great coral for associates. Makes a great home.
Okay, I'm listening. Okay.
Nicely done, Jake.
Alright, come wide.
Do we know how long is this coral?
Daniel, you guys still
It's over a meter long.
Over a meter long.
Yeah.
Say it again, Jake?
Did you get a WCA shot of this?
Yeah, I did.
Okay, awesome. Yeah.
Thanks.
Just checking. All right.
What can I do? For what? Oh, yeah. Oh, cool.
So if you are just tuning in now to the live stream, the coral we just saw is a little bit over a meter in length, and the two green dots you see on the screen is roughly 10 centimeters apart, or 4 inches, and that is what the team uses to help get an estimate of the size of the different species and animals that we see in the deep ocean.
Yeah, let's take a quick snap zoom there.
This
Okay.
is a glass sponge we're going to look at in the family Farreidae. This is called Farreid.
Push in there.
So it's definitely a Farreid, and we call it near oca because it's kind of like Farreid oca, but it's not quite. I like this one because it looks like pasta.
Yeah.
You're like, I'm ready for some mac and cheese. I think that's good.
All right.
Cool.
We can come back, White Aaron.
Yeah, let's do one of these sponges.
Okay. All right. Push in there.
Okay, so this one is Postolastra, for sure. Kind of like that yellow one, but this is like typical Postolastra.
Awesome.
So this is actually a demisponge and not a glass sponge, meaning that its skeleton is made up of more than just glass pieces. It has other types of materials that
That's
it
great,
uses
Jake.
to create its
Okay.
skeleton.
Come on.
Yeah, these things kind of are curved with the current, and so it helps funnel water through like that sieve of the sponge for better filtering. Oh, this boulder looks exciting.
Yeah.
Yeah, it does.
I think this is a good indication when it's about to come.
So exciting.
So all these white sponges you see, the tall ones, are Farreid near Oca erecta. We call them Oca erecta because they are tall instead of that short one that we were just looking at. There's also Calyptrophora. That's the primnoid that's in front of the purple coral. The purple coral is Iridigorgia alba, and it has a snake star on it. There are also some Chrysogorgia, some Dunlop sponges like Postolastra. There's like this dead stalk over here on the left side with the yellow bit on it that I'm interested in.
Okay.
Yeah.
So push in there.
Okay, so the yellow is a dema sponge. There is a small shrimp called Lebius on there. This shrimp is often associated with sponges. A bunch of zoanthids. There's a hermit crab. There's lots of foraminifera growing all over the place, as well as hydroids.
What's the purple?
The purple is the Victorgorgia.
They were seeing quite a lot of those on the previous watch as well.
Yeah.
Iridigorgia is.
Okay.
The purple and yellow makes a very striking combination.
Come a little bit wide there.
I love the zoomed in pans. They're very fun.
Bring up a still shot.
Yeah, there was also, I think, a madrepora off to the right. You can see it right in the corner of the
Okay.
Yeah, we're getting some beautiful photographs there
Here?
in the
Yep,
cinema
that's the
camera.
one.
That's fantastic.
So this coral is a scleractinian coral called madrepora. It is a deep sea reef building coral. So sometimes in certain areas we see this coral growing in large mounds, and it can make a great home for a lot of different types of invertebrates and fishes. Here we're seeing a squat lobster in its branches, along with a bunch of hydroids growing all over. I see also some ophiuroids, brittle stars, and possibly a mysid shrimp or amphipod. There's lots and lots of life.
All right. Come wide.
I think those little white things are stylastrids. Did you see those, Daniel? The little small sticks?
You mean these things there?
Yeah.
Yeah. Too far away for me to wager a guess there. But yeah, might as well take a quick peek.
All right, want to go in there for a quick?
Yeah, I think so.
Yeah.
I think they're stylastrids.
Hard to tell, yeah.
Okay.
They look a little bit fuzzy, so it's sort of indicating some polyps to me.
Yeah, it could also be bryozoans, but it's tough to say. But I have a feeling that pretty much every rock is going to have tough as we keep climbing.
Okay. Come on.
Yeah, a couple here for our viewers at home. Now Megan mentioned the reef building corals. So in shallow water reefs, and particularly here in Palau, we have several hundreds of reef building corals. In the deep sea globally, we only have about half a dozen species of corals that are known to form reefs. Two of which we have seen on this dive, the NA167 and madrepora. And there's also actually reef building deep water sponges. That's also been reported in Alaska, of British Columbia, and basically of the western coast of Canada and the United States, and not elsewhere in the Pacific. Yeah, but we're seeing quite a few glass sponges, and I have a feeling that we'll see more and more. Take a quick look at that black coral.
Which one? Sorry, I was thinking of.
It's in the upper left corner of the screen.
Oh, okay. Sorry, I was taking a sip of coffee.
It's a bit like lilipathes, but
I think Trissopathes.
yeah, we'll see here shortly.
Gonna come down on the winch a little bit, 5 meters or something. All right, you can push in there.
Yeah, I think you're right, Megan. As per usual. Yep.
Well, you know, when you do your taxonomy papers, you've got the thing in front of you, and I never have it in front of me. I'm always looking at it from really far away, you know, squinting my eyes.
Yeah.
Okay. What?
I usually look at how the branches come together. Trissopathes tends to be more at a closer to a 90-degree angle, whereas Lilipathes has a little bit more of an acute angle where the branches meet.
Yeah, thanks. And I think I'm also trying to like just think about because we'll see a lot more and more. And I think we're going to get overwhelmed. So I think what I'll try to propose here to the group is once we see something a few times to collect it, unless there's something incredibly unusual. But I'm going to guess a lot of this stuff is with the exception of the few things that are very, very well known. So once we see a few of the same that we haven't documented on this expedition, we should try to grab a sample.
Yeah, I think if we see that yellow Dema sponge that was forming a plate, that might be a good one to collect.
Have we collected any samples so far in this dive?
Yeah, we have. We've collected eDNA water on the bottom. We also collected some near some of this sponge habitat. We got a carbonate rock, and we've got a sea cucumber, a long purple-white sea cucumber, an encrusting zoanthids on an old coral skeleton, and a primnoid.
Fish. Cuskeel, maybe?
Yep, you got it, Jake.
Outstanding.
Push in there.
Well, this one has a little parasite on it.
Is that the little white thing?
Yeah, it's a leech, a little white thing on its chin, kind of jaw area. We often see the fishes with parasites. Oh,
There you go. Get a steam shot.
I wonder if we'll see that Coelurynchus that we're looking for.
That's great. That's
So a viewer is asking, what do cusk eels eat?
They eat small
Jake,
invertebrates,
can we take a quick look
and
at
they could eat small fish.
bottoms?
Yes.
Coming in center. Just
All right, quick snap zoom there.
Parantopathes.
yep. Wonderful. Thanks, Jake.
All right, come back on.
Oh, there's a calificus. So this is a glass sponge as well, in the family Roselidae, called calificus. The way to tell that it's a calificus is the long stalk that leads to this sort of mushroom head portion of the
Pushing a bit
main
there.
sponge body.
Nice.
And you can see some little amphipods acting as associates here on the sponge.
All right. All set there?
Yeah, good.
Good.
Aaron's like, I got more zoom. You can see the eyes
Go
on
all
the
the way in,
amphipods.
Yeah, beautiful imagery.
Nice.
Jake, seems pretty still, right, in terms of current? I haven't really picked
Yeah.
up on anything.
Yeah, no, nothing. Nothing really pushing the vehicle around.
Yeah, I'm really intrigued to see how that changes as we get to the top of this ridge. Another nice primnoid coral there.
That's a Calyptrophora. Oh, there's a little fish, a rat tail.
Yeah, maybe first on the fish and then on the yellow.
All right. Push in there. Yeah, just gonna keep moving by. Okay.
Excellent. Jeff, who's watching downstairs, just confirmed that this looks very much like one that I recorded previously, likely Kumba.
I love the name Kumba. It's cute. So this yellow coral is an octocoral in the family Acanthogorgiidae.
You have more zoom, Aaron? Okay, nice.
As we zoom in, we can see the polyps, and that'll help us narrow down what genus this might be part of.
And remind me, Megan, you were mentioning there's been some grouping there with the plexaurids, is that right?
Yeah, so the plexaurids have been split. So Plexauridae is still a family, and the Swiftie are still in the Plexauridae. But the yellow corals that were in the Plexauridae had been moved out into Paramuricidae, and now those corals have been moved into the Acanthorgorgiidae. So they've there's been a lot of rearranging the last couple years.
Yeah. Wonderful.
Right.
Thanks, Jake. And
Yep.
yeah, just take a moment here to acknowledge our very loyal and faithful scientists ashore. I really appreciate folks tuning in at all hours of the day. So right now we have on the chat Aaron Judah from the University of Hawaii, Sako Matsumoto from the Chiba Institute of Technology in Japan, Chris Kelly, former University of Hawaii, Ikelau Oto from the Palau International Coral Reef Center, James Reiner, University of Ryukyu in Japan, Jonathan Wambach, independent researcher Kenneth Sulak, formerly with the U.S. Geological Survey, and Steve Vaskovich, National Museum of Natural History, and Val Brown from the National Marine Sanctuary of American Samoa. Thank you so much for joining us. And yeah, we'll try to make some headway here. It looks like we are getting very close to waypoint 2. I have a feeling that that stretch between waypoint 2 and waypoint 3 is going to be an intriguing one. So I think at that point too, we'll notify our data engineer here, Michael Abriola, who has been working on a whole bunch of issues with the camera system, but we'll try to get a transect with our new wide field camera array, the cinema camera bit. So we'll try to do that here, and we'll let Mike know when that happens, probably in about, I guess, 20 minutes or so when we make it to waypoint 2.
There's a lot going on that branch, dead branch.
Yeah.
When we zoom in, I think those are carnivorous sponges on there.
The lower ones, the lower sponges.
Yeah, those little ones that look like lollipops.
Yeah, or cotton swabs.
Oh, here?
Either one, or the ones that are
I see.
higher up.
Yeah, I see. No,
Might be easier.
All right, push in there.
Yeah, so these are carnivorous sponges. They're Demis sponges. They kind of work like Venus flytraps of the deep sea. They will capture live prey, usually small crustaceans, things that are swimming through the water, and then slowly dissolve them in the similar fashion of a Venus flytrap. We also have a Crotella metra, a type of crinoid or feather star. There are also some gooseneck barnacles that have like very, very thick goosenecky texture. And then a Brisingid sea star called Novadinia. I believe this is Novadinia pacifica. And the yellow coral further down is Acanthogorgia. There's also some stoloniferous octocorals. And on the Novadinia, I'm spotting a polychaete worm. It's very cool.
Yeah, wonderful. Thanks, Jake.
Yep.
Hey Megan,
Yes.
we have a question about those last corals that we saw, the yellow ones.
Uh-huh.
They were asking why did they change its classification?
So there was a lot of work done using DNA analysis, and that showed the scientists that certain animals were actually more closely related to each other than we originally thought. And so they've been rearranging the octocorals to better match the findings of the DNA analysis.
Cool.
Originally, when the taxonomists were looking at these animals, they grouped them based on their morphology. And so certain scientists are lumpers and certain scientists are splitters. Some people like to group
Jake,
things together that look very similar, and
Yep.
other people say, hey, this thing is different, it should have its own group. So
All right,
throughout
go for a
taxonomy
zoom.
and history of classifying Earth's biological organisms, we've kind of got a lot of things that we've classified really well together, but other things are a little more cryptic and can be more challenging. And with advances in technology and our understanding of DNA and how animals are related, we've kind of started to update the taxonomy to reflect these findings.
That's great. Thanks, Jake.
Okay.
This is an anemone in the family Actinostolidae. These ones can get pretty big. I like them. They're fun.
First ever ROV sample I collected was
One of these?
Yeah.
That's fantastic.
the arm. It came up looking a little hourglass shaped.
Yeah.
That's great.
Like an anemone corset.
Yes. Yeah.
Let's take a quick zoom on that white coral.
Down here?
Yeah, the lower one. Yeah, that one. So this is a primnoid. And when we zoom in, we'll be able to see the polyps, and that'll give us a better idea of what genus this belongs in.
All right, push in there.
So what I'm looking for is which direction the polyps fold down in, if they have 3 tiers of scales or more, and how the coral is branching. So I think this might be a Calyptrophora that's pretty young, but it could also be a Narella.
Yeah, I think, Chris also thinks it's an Narella.
Yeah.
Thanks. That's great, Jake.
Okay.
And there's also a snake star in its branches.
Oh boy. Is that a hemicorallium?
Yeah, it
Yes.
is.
Maybe let's take a quick zoom on that. That's new for this shift, at least.
I think they were seeing some like really white hemicorallium earlier. This one is pink. They're usually pink,
Yeah.
but they can vary in color from being a very deep color pink to almost pretty much fully white. And there's also a rock pen called Athoptylum.
Alright, you can push in there.
And on this coral, we have brittle star as well as probably an amphipod hanging out in the branches. The way to tell the hemicoralliums
That's
from
great. Thanks,
the
Jake.
Paragorgia
Okay.
are that the hemicoralliums have paired polyps at the ends of the branches.
Megan, do you have a sense of range to waypoint 2?
I think it's about 60 meters. Let me check. 80 meters.
Roger, okay. I just want to give Mike proper warning. Yeah, another Trissopathes there, another Chrysogorgid. It's quite, and actually can we just, I want to just general zoom in here.
On the sediment.
Yeah, just kind of sense of what the texture is there.
Yeah, it might be a place where the manganese crusting is broken off.
Alright, you can push in there.
Oh, you can see a fossil.
Oh, yeah.
Yeah, so this is old reef.
Yeah, that's great. Thanks, Jake. Yeah, this is exactly what I wanted to confirm. Yep, perfect. So this is a good, thanks, Jake. Yeah.
Okay.
Reminder that this was once a shallow water reef built by reef building corals, crustinous coralline algae, and various other algae that cement together this structure. Those are structures that typically form at 100 to 200 meters where your light still comes, and obviously we're quite a bit deeper here, 1100 meters, so this was once much shallower and has since sunk to these depths.
Oh yeah, let's look at that one.
Yeah, let's take a quick gander.
So this coral can be one of two things, either a white Paragorgia or a Subogorgia.
The bigger one?
Yeah, the big one.
Okay.
It's kind of different from the other things what we'd been looking at.
Yeah. Alright, you can push in there.
Yeah, unfortunately, the way to really confirm what the genus is for this particular coral is to look at its sclerites. Those are little hard pieces in the tissue that are microscopic. So we can't really confirm that from this angle with this camera. But it's great to get a good look at
Yeah.
the coral itself.
Aaron doesn't have that much zoom.
So what were you saying, Megan? This is Pleuroccorallium or Subagogorgia?
I think it's Subagogorgia or a white paragorgia.
Got it. Okay.
It's definitely not a Pleuroccorallium.
Got it.
Because you see the rounded ends of the branches and that sort of yellowish tint to the base indicates that it's not your hard calcite corallidae skeleton.
Yeah, wonderful. Thanks, Megan. And
Okay. Happy here, you can come on.
it's like Chris and Matsumoto are frantically typing, but yeah, Sako is also agreeing that it's hard to put an ID on this one. Wonderful.
We have a question for Jake.
Sure.
What is the maximum speed that the ROV can operate in?
We can go about 1 meter per second, or 2 knots, forward, and then laterally, not as fast, maybe a quarter of that speed. It's a lot harder to move laterally. We don't have as much thrust. The lateral thrusters are smaller. But moving forward, generally, we can get about 1 meter per second. But right now, for example, we're moving pretty slowly. 0.3 knots. The ship's moving, so ROV we're tend to just follow that speed and move along slowly with the ship. Any faster, and it's very hard to identify things on the bottom, and you tend to miss a lot of things.
Here we have some treated pleura glass sponges, and then there's a sedarid urchin. Yeah, that's pretty cool.
Oh, the spiky?
Yeah, the spiky thing.
Yeah, for those that are following along with the dive plan, so I think we've made it about 400 meters of horizontal ground that we've covered over the last several hours. There's still about 60 meters or so till the top of this feature. We've set out a plan that basically goes in an inverted L shape where we first climb up the slope, and then there's a narrow ridge at the top that will start falling parallel to the ridge. And I have a sense that, yeah, once we get to that top of that ridge feature, we'll see a lot of action, even more action than we're seeing already. Maybe quick peek in here. Is that a dead sponge or another black coral? Let's take a quick look.
Nice focus on the WCA.
Yeah, spectacular. My partner in crime here, Makani, has been taking some really, really nice photos. So big
Nice
shout
shot.
out to the large team that has been working to get this camera up and running. We've been have several folks behind the scenes. Our data engineer, Michael Labriola, our ROV pilot, not on shift, Dan Cormany, and Megan Cook. Several have been working to streamline the processes here, and yeah, we've been getting some really nice shots here. Yeah, we can just take quick zoom on that. It seems.
Can push in there.
I think it's a dead coral with hydroids on it.
Yep. Wonderful, that's great. I just wanted to confirm.
Come back wide. Yeah.
Yeah, let's
Oh
zoom
yeah,
in on that.
the little
Yeah, let's push in on
homelid
the
crab.
Push in on the crab, please.
This is a carrier crab that always carries an anemone called Lamoha. I mean, it's a really fantastic hat. Height of fashion. I'm jealous. I know, right?
That's wonderful.
Nice. Okay. And come on, come back wide.
Yeah, nice discussion going on here in the chat by our scientists ashore, comparing some of the things we've seen to things that have previously been seen by other efforts that have worked in the Central and Western Pacific. So there have been quite a few previous expeditions in and around the Hawaiian and Pacific remote, U.S. Pacific remote islands, and also the Mariana Islands. We are here in a place where we haven't really explored with this kind of technology. There have been several dives using JAMSTEC's human-occupied vehicles, but quite a bit deeper. Those explore depths of 6,000 meters and deeper. And so, yeah, one of the longer-term goals of this effort, in addition to trying to just map and explore and characterize the Palau National Marine Sanctuaries is also to combine the data set that is collected on this expedition with those that have been collected previously and will continue to be collected throughout the Pacific. The study at biogeography, the study of how species are distributed, that's relatively well studied in shallow water and deep water. There's been a proposal of the biogeographic provinces. Dr. Les Watling put on a really nice paper about 10 years ago trying to group bigger areas in terms of what kind of fauna we expect
zoom on
there.
that sort of polka dotty one?
But it has not really been ground truth much.
Oh, down low?
Yeah.
To the right?
Yeah. I think it's a soft coral.
Push in there.
Oh no, it's a sponge full of zoanthids, but it's a dead sponge. That's pretty cool.
Yeah. So many.
I wonder if they were in there before the sponge died or if they colonized afterwards. They like to be very equidistant. We often see sponges colonized by anemones or zoanthids while they're alive.
Could the number of them, would that be a reason for the sponge to die?
Possibly.
It just got taken over by all the zoanthids?
Yeah, too many and it wasn't able to feed.
Yeah.
Yeah, but that's an interesting question because that's in several places. It's been, you have these massive aggregations of something growing on something that is dying or dead, and then you get the chicken or the egg question. But in essence, every organism, once they die, they become substrate for a whole bunch of things, especially for things that like to need that hard ground to attach to. I'm personally very interested in this glass sponge that there's also a species of black coral that has been described only to live in that, but I haven't seen it yet.
Oh, I did see one, so hopefully we'll see another one. So there, on the farreid sponges, most often we see this it's an antipathes, right?
Yep.
Antipathes spilospongia.
That's correct. But it's probably going to have to remove from that genus. Antipathes is kind of considered a taxonomic dumping ground.
Yeah.
When black corals were first described, that was the first and only genus, and then more and more things got added to it, and then only over the last 20 years or so have new families in general have been erected to try to classify them in a more in a way that makes more sense.
I think I see a Rhodaniridogorgia in the upper camera.
Okay, come wide a little bit.
Yeah, it's...
Oh, off to the...
Yeah, it's right. Off to the right there.
Off to starboard there.
There it is. Actually, it might be a iridigorgia. See how it has a deeper spiral? But this one's branching, which is a bit unusual for iridigorgia. And then in the branches, I see a shrimp called Bathypalamonella. There's actually a couple of those shrimp in there.
What's that little thing just below the shrimp?
Oh, I think that's eggs if we can zoom in. That might be something Harold will be really interested in.
You can push in.
Yeah, that's definitely eggs. Probably fish eggs. I wonder if Harold's watching upstairs or downstairs.
Yep, there is a request to please collect, so I think
Yeah.
if we're set up to do so, Jake would like to go ahead and do that.
Okay. Come wide.
That's going to require
The
probably
ship is stopped.
clipping
Nice.
that right at the base.
Harold's been really interested in the association of fish and corals, and time and again we've been seeing eggs laid on different coral species, and we kind of want to resolve more about how that relationship works.
Yeah, Jake, if we can just maybe just cut right underneath the egg so we get a piece of the coral and the eggs, that would be ideal.
These types of corals tend to be pretty floaty. This is also a good collection because this this coral itself is a bit unusual.
What do you mean by unusual?
You can push in a little bit there.
So usually
Tilt the camera
iridigorgia don't branch, and this one definitely has branches. But
All right.
the
Push in a little bit more. All right, just want to make sure I see the.
the Rhodaniridogorgia will branch, but they don't have as deep a spiral on the main axis of the coral.
Mm.
So it could be something new or something I'm not fully familiar with. I know Les Watling's been working on the taxonomy of this group. I think you might need to get a little lower. Yeah. That might be good, yeah. Looks good.
I might get some other pieces with it, but there's nothing I can really do.
Yeah, well, this coral's very all over the place.
Yeah, I got multiple pieces there.
Alright, so if you want to put it in the forward box,
Okay.
you can. Maybe that's easiest.
Yeah, starboard would be hard with the thruster.
Yep.
Yeah, so
Camera.
forward omega, or the starboard side of the forward box.
Roger that. Forward, starboard. Yep. Tilt down.
And Megan, this is 81.
81, perfect.
And then down my. Oh, it's already on. All right, can bump open the box.
Oh, we have the scoop in there, don't we?
Oh yeah, the scoop's in the starboard side.
You can just put it in the port side because there's
Okay.
a sea cucumber and those are very clearly different. So
You can bump it out a little bit more.
just in case we need the scoop.
bump it out a little bit more. Oh. Fix the box there. All right, close the box.
Nice. Well done.
Yeah, great collection. Big thank yous here in the chat.
Oh yeah, a little. Yeah, you can bump it up a little bit. Oh, that's a little hot.
There's like that special threshold. You know, it's
Totally.
either all or none.
Yeah, fish there at bottom of the screen.
Go for a zoom.
What is it, Jake?
Cuskeel. I have about 3 identifications down. That's one of them.
You got to start somewhere.
Yep.
You're doing
Work
great.
your way up. Yep. All right. Ship stopped?
Ship is stopped.
Okay.
Just giving you some time to
This looks
catch
like
up.
this looks like a decent wall.
Oh yeah.
With some
Yeah, nice view here to the left, and I think this is kind of what we're going to start seeing more in a lot of these sponges.
Mm-hmm.
A lot of these yellow crinoids perched on top.
These crinoids are Crotella.
Yeah, so we should be pretty close to the...
Oh yeah, we're super close. We've got about 40 meters.
Yeah.
Oh, there's more of that
Yeah.
yellow sponge.
Yeah, I think this is what we were considering collecting,
Yeah.
and we see now several of them, so I think if we're set up to do so, Jake, yeah, maybe grabbing a piece of one of these yellow sponges.
Sure. See
Nice.
what I can do. You can come up a little bit, Sarah. Like maybe 20, 25 meter delta.
And I see several of these white mystery corals, but I think there was a little bit of a convergence towards actually pleurocorallium and the chat.
See if maybe this one will be easier.
What is the yellow sponge?
It is a demisponge.
Demi sponge.
Mm-hmm. So there are three main groups of sponges. You have your glass sponges, your demisponges, and your calcareous sponges. And so this is the demisponge. We call them those names based on what they use to make their skeletons. So the glass sponges use exclusively glass, but demisponges can use a variety of materials and multiple different types of materials.
And they would be smooth to the touch as opposed to the more glassy and sharp feel of glass sponges.
Oh, there's definitely been some really spiky demisponges I've touched.
Yeah.
They can have long glass spicules in addition to, like a more soft ocean sponge that you might have at home kind of texture. So all the sponges that, are ocean sponges that you might, have bought at the store are demisponges.
Oh, man.
Oh, maybe this isn't a good spot.
Yeah, it's a bit of an overhang. I tried to land the...
Yeah, well, I'm sure we'll see some, Jake. No.
I almost had it.
Yeah, no worries. I'm sure we'll see.
Slid right off.
Seems...
I see there's a fuzzy looking sponge. I think that one has the
Just looking at that,
Antipatheria
left
on it.
left us. Which would be, we'll try to get a good zoom in.
Oh, wow. Yeah, that is a boulder.
All the action all around it.
Yeah. Any area of elevation is going to be taken advantage
Come
of.
down a little bit now, maybe 5 meters. Get bounced around.
So there are some Narella primnoid corals, hemicorallium, pink coral, madrepora, hard coral, chrysogorgia, as well as a ton of these glass sponges
Yeah, and Chris
that are
is
farreids. Is Chris in the chat?
yeah, and he
Yeah.
is remarking that we should try to collect one given their abundance and how representative they are of the environment. So maybe in a place where it's easier, because yeah, they're just so abundant. I think we can try to find a rock where one is a bit more isolated.
Yep.
Chris, do you think that these are Farreid near Oca erecta, or are they different? Because the way that they're shaped is more like little cups than like tubes on the sponge. Looks a bit different than what we normally see.
Yep.
Yeah, Jeff is also remarking, yeah, so we're going to like try to find a place where it's not under overhang, and we'll try to sample one of these farreids.
Nice.
So yeah, and Chris is actually thinking it might be a different genus, so we'll try to look for one where we're not in this dense bed where we'll likely to bump something, so we'll look for a rock where they're more isolated and try to sample one. Yeah, this is exactly what we were expected, and we're getting closer to the top of the ridge, and big boulders here, a lot of dense aggregations. Yeah. That's Venus flytrap anemone there at the bottom, many primnoids, many glass sponges,
Where do we start?
bubblegum corals, dead sponges. Yeah. Yeah.
Let's look at this big Venus.
So this is Pheliactis, a anemone in the family Hormathiidae.
Push in there.
It has a little shrimp friend. Looks like it could be a Lebius.
Yeah, and if Mike, our data engineer, is tuning in, I think we're pretty close to wanting to start a photo transect to take periodic stills. So we're going to try to do that here in a couple minutes. We are almost on site where the ridge. So hopefully, Mike, if you're tuning in, and let us know if you're ready.
So
All right, wonderful.
Is that a sponge with a bunch of those?
should
thanks.
we,
Yeah.
Thanks, Mike.
That's a lot.
Daniel and Mike, are we just going ahead and starting it on our end, or
I think
Push it
once
to
we're done here, we can hit the
sweet?
Come on.
top, and then we'll just try to,
Great.
and yeah, maybe just make a note in the,
Yeah, definitely.
perfect. Yeah,
On the video transect, are we doing a true transect, keeping a height above bottom and no stopping?
no, just
No,
we're going to do the kind of the photogrammetry transect here between waypoint
Okay.
2
Fly
and
around,
3.
get a bunch of different angles, get a, yeah.
Yeah, so just feel free to fly as you want, Jake, but yeah,
Yep.
we'll just basically activate the cinema cam to take a photo every 2 seconds.
Okay.
And yeah, we'll try to keep the autofocus on, and
Yeah,
then-
I think talking with Megan, we'll either keep the autofocus on or sort of tune it a little bit as you go. 'Cause the
Okay.
autofocus looks like it's really out.
Okay.
So
perfect.
whatever capacity we have to, it's
Perfect.
too hard to tune every 2 seconds, but
Yeah.
Looks like there's some of that
yeah.
yellow
Perfect.
sponge we were trying to collect over there. Maybe that's a better spot.
So, Connie, if you want to, whenever you're ready. yeah, you can just hit the toggle timer and then we'll start. There we go, and we are
All right.
underway.
Survey start.
Yeah, thanks. And yeah, I mean, we can adjust the focus if you think it's getting off a little bit, but yeah, maybe let's keep the autofocus on too. And then you can, yeah, fine tune it, as May was saying. Yeah, this is a wonderful, yeah, there's a piece of sponge that almost looks like it's already loose, or is there?
Oh, the yellow one?
Yeah.
This is really cool. How long does it take for some of these really big sponges to grow to this size? Do you know, Megan?
We actually don't know. There's no good way to date sponges. Obviously, they grow slowly, but it's hard to get an accurate understanding of their growth rates because they don't have good ways for dating them. Some of the demo sponges might be able to be dated, but the glass sponges are not.
Yeah, Chris Kelly also wrote in and said no one knows. So yeah, that's interesting.
That's unlike a lot of the corals that we see here, which in a lot of groups they form growth rings, just like trees, terrestrial trees would do.
Right.
So you would cut the base and cross section and look under a microscope, and then count the rings, and those rings are not necessarily years, but there are periods of faster and slower growth, some kind of regular cycles. But then also for a lot of these really, really slow growing corals, you have basically using bomb carbon to date them. So in the late 60s, there was several tests of atomic bombs, and those materials, those radioactive, were taking on by the tissues, and then basically if you have a sample, you can measure inward from the outside, and at some point you will pick up that bomb carbon signal, and you can measure then how far in that grew, and that gives you, from present day to the late 60s, and then you can extrapolate to the center and get an estimate of how much time has passed. Several other methods is obviously when these corals or sponges grow on human-made artifacts, on shipwrecks or other things, you know that they can't be older than when that wreck went down. So several different methods have been used to date. And in a nutshell, the key thing to know is that a lot of these deep sea species grow a lot slower than their shallow water counterparts.
I wasn't familiar with the bomb carbon dating. That's really intense. Is there other species that in the deep that use a similar method that have been affected and sort of trace that signature?
Yeah, so there was a pretty key paper by Dr. Bren Rourke at Texas A&M University, who's been very interested in the growth. He dated 3 different corals in Hawaii using this method, black corals, some pink corals, and bamboo corals, I believe, also, and got order. The oldest were on the order of closer to 4,500 meters, 4,500 years for a species of black coral, and some others were in the order of several hundred years. As I understand, he's working on a few other species where he's gotten rates and over 10,000 years, are not yet published for some species, and so yeah, it's fascinating. It's obviously difficult to get samples, and
I guess there'd be certainly some level of uncertainty in terms of periods where growth would be faster or slower, and so there's maybe like a range that he kind of offers for these.
Yeah, absolutely. And I mean that's true for most organisms. Is just like us humans, we have a period when we grow very fast, and then at some point we stop growing. And it's a common phenomenon in a lot of organisms. Is when you first are born, a lot of energy goes into growth, and then at some point the energy swaps over and goes into reproduction. But also in the deep sea, in particular like sponges or so, you don't have a phenomenon called apoptosis or programmed cell death. So a lot of organisms, the cells would at some point die. And a lot of these organisms don't have that phenomena. They could, in essence, live forever. But what happens actually is in some cases, especially for some of these massive corals and sponges, at some point they will become so massive that they become too big for the holdfast to keep hold of them, and they'll just topple over. And you'll see this throughout these surveys, is that you'll see these big, both corals and sponges that have toppled over. In some cases they can reattach, but in others they can't, and it's a function of how much current there is. And
which direction do we want to move? Do we want to move along this wall feature, or do we want to keep?
Yeah, I think so. If we can keep kind of at the...
I've gone back and forth on this wall twice,
Yeah.
so yeah, if we want to move in a
Well, we've got...
direction, is this like part where we're going to make our L-shaped move?
Yeah, so we could start heading towards waypoint 3 right now if we want, or make another move to officially get to the marker for waypoint 2. What would we like to see?
Yeah, I mean, I think this seems pretty... We're here at the edge, right, of the... Like if we drew a point now towards waypoint 3, would be pretty much along the edge of the... So I think we'll want to move along that and then maybe periodically just peek over.
Okay.
And hold off on that sponge collection?
Yeah, well, I think I want to just get where we feel we can reasonably put the ROV down, so we're not setting down a lot of things, and
It's quite dense.
Provide, avoid
on a wall.
any. Yeah,
So,
exactly.
not many landing spots.
Yeah, and in addition to that yellow sponge, I think that white sponge that's also very common, I think I will also want to try to sample at some point.
Yeah, the farreid.
Yeah, exactly. Or yeah, but this is a...
All right, so I'll move us north 010.
Okay.
Yeah. So yeah, at some point, not right now, Jake, but we'll want to collect one of those.
Yep.
And then the bright yellow one, but whenever we see one that we could collect without any collateral damage.
Yep. What was that move direction?
010.
010.
It seems like this wall is pretty north, but I wanted to bring Atalanta in a little bit.
Yeah.
Yeah, on the cinema cam we have just a spectacular view of the top of this feature and how much life there is. Just a perfect illustration, I think, for most people. When they think about the deep sea, they think about this emptiness, this void of life, and here is a perfect illustration of that misconception. And if you look in the right places, there's a lot of life.
And even in the places where it looks like there's not life, there's a lot of life that's very tiny.
Oh, yeah.
So pretty much everywhere there's a lot of life. It's
Yeah.
just on what scale.
Yeah, and how you look for it.
Yeah.
Exactly.
Yeah, that's a really good point. And for the microbiologists, they always point out that those are the real bases of food chains, and that's where the action happens, and the microscopic life. You know, those are so important for nutrient cycling and so many things. And every one of these big sponges and corals, if you were to quantify how many microscopic organisms are on them, they would be on the order of billions. Does that look pretty much in focus? Yeah, great. Yeah, that's wonderful. So yeah, we'll try to move along here. For those that are following along and have seen the dive plan, so we started the dive basically moving west to east, and we did that covering about close to 500 meters, and now the rest of the dive is going to start moving more in a south to northeast direction, moving parallel. This seamount has a very unique feature in that it's in essence a long narrow ridge. Yeah, a lot of primnoids, a lot of these glass sponges, hemicoralliums. Acanthogorgids. Yeah, this is wonderful. This is our second, what we would really, without any doubt, call a higher density or high density community. We had one on the very first dive of the expedition, also at similar depths. And yeah, looking ahead, tomorrow's dive is likely going to take us to somewhat deeper depths, although we'll get there. And then on the first dive of the second expedition, which will be November 11th here in Palau, that is a dive that I'm particularly excited about, because that is a seamount that basically stands all by itself. It's in the very southern portion of Palau National Marine Sanctuary. It's at the depth of probably 500 meters or shallower. We don't yet know exactly because that seamount hasn't yet been mapped, and it's completely out in its own big seamount.
Is it wrap? Okay.
What's that in the water?
Oh, I don't know.
That's a shrimp.
Push in there.
Yeah, it's
Oh.
a shrimp.
Is it shrimp? Yeah.
Oh, there's our heterocarpus. It's a big shrimp on the rocks.
Yeah, we'll continue moving here along. Two things that I think we'll definitely want to collect whenever we're in a position to do so is one of these white farreid sponges and one of those bright yellow demo sponges. I have a feeling it's probably once we get to the top of this ridge, it might be a little bit easier to find that yellow one there. That's the one we're looking out for.
Yeah, the yellow sponge could be something like Mike Halley, but I'm not sure.
Huh, I'm used to Michaelia like being a really shallow water genus.
Yeah, usually. But it kind of looks similar in many ways, but the demo sponges are a particularly difficult group. A lot of things look very similar. Again, you're looking at the skeletal pieces inside the sponge skeleton to be able to identify them to species. Oh wow, look at that bolosoma. That's pretty cool. So this big sponge coming into view is a glass sponge in the family Euplectellidae called bolosoma. It might remind you of the one that we looked at called Caulophacus, but the way to tell the difference is how that stalk attaches to the base of the sponge, and it has these two large holes or oscula, one on the top and one sort of facing towards the ROV.
And so why does it have the extra one?
Why does it have two holes?
Yeah.
It's just how the water funnels through the sponge. That morphology works for that sponge. And there are many ways sponges move water through their bodies, and they all have very, very different shapes. But that shape tends to be a good way to funnel water through that sponge body.
Do you think this jake is in an isolated enough situation to collect there? Yeah, I'm just trying to... Either that
Maybe
or
this
this.
one right
Yeah.
here. Yeah. Could definitely get this one.
Yeah, might as well before.
Okay.
So this sponge is going to be very fragile and break apart very easily.
Okay. Is it a whole
If you grab the whole,
sponge collection, or is
yeah,
it
if you
gonna...
grab the whole thing, you could probably stick it in the starboard box. Otherwise, we could suction it. It'll fragment.
a fragment? Well, I guess that's up to
Yeah.
science of whether we want to try to collect the full.
If Chris is in the chat, maybe he can weigh in about what might be more ideal for collection. A larger whole piece or just small pieces are okay?
Yeah, he's typing in here, is also mentioning that the sponge right next to the one that we're about to collect is a farreid. Yeah, so you said about a fourth of it, but yeah, I think probably if we clip it, I would think, as opposed to suctioning it.
Yeah. Okay.
So yeah, maybe the distal quarter of it.
Push up.
So clip it and put it in starboard box, or clip it and slurp the clip?
I would put it in the starboard box. Otherwise, it's just going to fragment into tiny pieces in the slurp.
Yeah, agreed.
Yeah.
It should be negative, so it should fall into the box easily,
Okay, nice.
but it's very fragile.
About a fourth of it, that's what you said?
Yeah.
Yeah.
Can we get a zoom on it before
Yeah,
you snip
We
it?
can push in here. Tilt the camera right a bit.
Nice. Cool. Thanks.
Okay. go for it there. Is that all right?
Yeah, looks great.
Oh, got the whole thing. Didn't break.
We'll take it if it fits.
I guess the main axis of the sponge is a little more tough than I anticipated.
All right, so the starboard box, the only thing we have in there is in B, the little one second to forward. So if you want to put it in one of the outboard bigger boxes, that would work.
Okay.
Or if it fits in a small one, can you do that?
All right, you can switch over to the sample salvo. Is there anything floaty to worry about in the starboard box?
No, it's just a rock.
Okay. You can bump out the box. You can bump out a little further. I think we're going to try for one of the smaller ones, right?
If you think you can, yeah, that
Yeah, I
would be
think
great.
so.
Nice. Yeah, so the forwardmost one or the two aft ones are available.
On the little top half.
Nice.
Okay, you can close it.
Great job.
Outstanding. I just had stopped here the camera, but we'll restart it when we'll move. Just so
Okay, noted.
yeah, just for a second. Didn't want to have 500 photos of the same.
Yeah, understood.
You switch back to... Nope. Still pieces left. Just gonna have to lift off here.
All right, just restarted the camera.
Roger.
Wonderful, that's a great collection, and Chris, who's been really dedicating his attention to sponges, mentions that he hadn't seen this type of sponge, and the fact that it's so dominant on this dive is a really good combination.
Oh, there's that yellow one.
Yeah,
Yep.
that's the one we also want to sample whenever we're in a good spot. We'll look to you, Jake, and where you think that might be. I think we'll see it commonly to wherever you
Okay.
think you're set up to do so.
Gotta put the arm in a little bit.
Yeah, I have a feeling we're going to see a lot more of these. So,
Okay.
so wherever we feel like we're in a good spot.
curve of the wall is changing.
Starting to, yeah.
It's starting to curve this way. So it's about 035 to waypoint 3. Looks like you're going 045 right now.
Yeah. That's my...
Looks like we're going to have our first unbranched bamboo coral, perhaps. It's going to be coming up here to our left. See it in the cinema cam.
That camera isn't actually streaming because it freezes pretty consistently when you're doing the capturing.
Yeah, thanks for that, Sarah and Aaron. Well, yeah, here we can just take a look at that guy.
Yep. Come down 5 meters. Push in there.
Ooh, this one has really long intertentacular spines.
Is this the Bathygorgia?
No. Bathygorgia is generally deeper living. I think this might be Lepidopsis. And usually Bathygorgia, all the polyps are on one side of the stalk. These ones are definitely whorled around.
Yeah, wonderful. Thanks, Megan.
Okay, come on.
Large paranthropathies, a lot of these sponges that we just collected, also hemicorallium.
I love the texture of that Rhodaniridogorgia. This one's going to be a challenge to annotate, that's for sure.
Nice madrepora underneath the overhang. Yeah, it looks like a lot of these farreids have zoanthids on them.
Yeah.
I do want to confirm the black coral, one of them, because I haven't really seen it. See the fuzziness from afar, but should try to get a zoom in when we see a fuzzy one.
Yeah, I'll keep my eyes out for that. Yeah, it seems to only grow on it doesn't seem to grow on the one that we collected that's been really abundant, but like the regular farrea. Oh. Okay, is that better? Okay. Oh, there's a sea star.
Nice. Do you want to look a zoom in it?
Yeah,
let's do
let's
that.
take a look at that one. So this is a goniasterid sea star. They are coral predators.
Push in there.
So this coral, this sea star knows what's up. This is a hippasteria. And it's probably making its way over to its next meal. We often see these sea stars
Nice
on
benthictinophores also on the
different
side.
corals. Oh yeah, there's those Gelfila benthictinophores on the sponge. Previously we saw them on corals, but they also seem to like sponges as well. And then this other sea star that we're looking at that's got the many, many legs is a brachyngid sea star called Novodinia pacifica.
All right, come on.
There's a black coral,
Yeah.
the Parantopathes.
Yeah. Yeah, well, it's a beautiful terrain here. So when you annotate, Megan, do you actually put a name on every single individual?
Yes,
Oh.
every single individual
This
gets.
one's going to take a long time.
Yeah, so, but in the cases where, say, we've got many of the same kind of thing, we can do a categorical abundance. And that means, say we see 50 of the same sponge in the same view, I'll just annotate that sponge once and count how just estimate the number that I'm seeing. So our categorical abundances start from 21 to 50, 50 to 100, and 100 or more.
I'm set up here to this yellow. This is the yellow sponge, right?
Yeah,
Yep.
that's the yellow sponge.
All right.
If you're set up to do so, that'd be a great
And the ship
candidate.
is stopped.
So we gotta zoom on it before we sample it.
Yeah.
Push in there.
Yes.
Yeah, that would be ideal
Okay.
if it's possible.
This is going to be soft and a little bit squishy.
Okay.
Should we stop the camera, May, you think? Okay,
Yeah.
America.
Yeah.
You
Yeah.
could pause it, yeah. I've been making a note when we pause it.
Perfect, yeah.
Yeah.
All right. Come wide of it.
But it should be dense.
So if I
But I've never
grab a
yeah.
big chunk of it, it hopefully won't.
Hopefully,
million pieces?
yeah. Hopefully, it won't